Imagine a tiny embryo, just 16 days old, setting the stage for the entire nervous system. Pretty incredible, right? Welcome to Neurology Embryology Part 1, where we are going to discuss the neurulation and neural crest migration. It is a must-know topic for USMLE Step 1. This process is foundation of how our brain, spinal cord, and peripheral nerves form, and it's packed with the high-yield points you will see on the exam. Get ready to connect the dots between embryology and clinical relevance. Let's dive into neurulation next. Picture this. A flat sheet of cells in an embryo suddenly starts folding into origami to create the nervous system. Pretty amazing, right? That's neurulation. And it's a high-yield topic for USMLE Step 1. Let's break down the essentials you need to know. Neurulation starts around day 16 post-fertilization when the embryo cells are organized into three germ layers: ectoderm, mesoderm, and endoderm. You can remember them with the mnemonic Easy Mamas Endure. Ectoderm, mesoderm, endoderm. The ectoderm is our focus here because it gives rise to the nervous system. Specifically, a portion of the ectoderm thickens to form the neural plate, which is the first step in building the brain and spinal cord. Why does this matter? Neurulation forms the foundation of the nervous system through neural tube formation. This tube will eventually become the brain and spinal cord, making it a critical process to understand for step 1. If this process fails, you get defects like spina bifida or anencephaly. Classic exam questions often linked to folate deficiency in clinical vignettes. For example, a question might describe a newborn with a lower back defect and ask for the embryological cause. Knowing neurulation helps you nail that answer. The neural plate doesn't form in isolation. It's induced by signals from the underlying mesoderm, which we will talk about later, and it sets the stage for neural folds and groove to develop. So, as we move forward, keep this big picture in mind. Neurulation is the starting point for the central nervous system, and understanding it is the key to tackling those embryology questions on the exam. Let's zoom into the embryo at day 16. Something incredible is happening. A flat sheet of cells is about to transform into the nervous system. This is the ectoderm's starring role in the neurulation, and it's a must-know for USMLE Step 1. The ectoderm gives rise to the neuroectoderm, which forms the neural plate, a thickened, flat, oval-shaped sheet of cells. You can see this in the diagram. A. The neural plate sits above the notochord with the precursor of the neural crest nearby. Here's a mnemonic to lock this in. Nervous ectoderm plates rise. Neuroectoderm to plate to ridges. The neural plate cells then replicates rapidly, forming two ridges called neural folds, with a depression between them known as the neural groove, as shown in diagram B. This process is critical because the neural folds will eventually fuse to form the neural tube, which becomes the brain and spinal cord. Diagrams C and D shows the progression. The neural folds rise and come together, pinching off neural crest cells that will migrate elsewhere, while the tube closes to form the neural tube. For step 1, you need to know that the ectoderm is the origin of the central nervous system, and defects here can lead to conditions like spina bifida, often tested in the clinical scenarios involving the folate deficiency. The ectoderm doesn't work alone. It's induced by the notochord, which we will cover next, but its role is foundational. So as we move forward, keep this in mind. The ectoderm sets the stage for the nervous system, and understanding its transformation into the neural plate and folds is key for those embryology questions on the exam. Let's imagine a tiny embryo where one structure acts like a director, telling the neural plate to start folding. That's the notochord, and it's a key player in neurulation. Let's dive into the mesoderm's role, a high-yield topic for USMLE Step 1. The mesoderm gives rise to the notochord, a tube-like structure that runs along the embryo's midline as shown in the diagram. The notochord is critical because it sends signals to the overlying ectoderm, including the neural plate to form the neural folds. You can see this in the diagram. The notochord sits beneath the ectoderm with the mesoderm and endoderm around it. The neural folds then grow around the neural groove as the notochord directs them to enlarge and eventually fuse into the neural tube, which will become the brain and spinal cord. This process is a big deal for step 1 because the notochord signaling ensures proper neural tube formation. If this goes wrong, you get neural tube defects like spina bifida or anencephaly. Classic exam questions often tied to folate deficiency or genetic factors. For example, a vignette might describe a newborn with midline defect and ask what structure signaling failed. That's the notochord. The mesoderm doesn't just form the notochord, it also gives rise to structures like the somites, which we will touch on later when we discuss neural crest formation. But for now, focus on this. The notochord is the mesoderm's key contribution to neurulation, setting the stage for the nervous system next. We will explore the endoderm's role and how the neural tube forms. Let's take a step back and think about the embryo's environment. While the ectoderm and mesoderm are busy building the nervous system, another layer is quietly supporting the whole process. That's the endoderm, and its role in neurulation is subtle but important point for USMLE Step 1. The endoderm forms the lining of the GI tract and respiratory system, which might seem unrelated to the nervous system at first glance, but its role is critical for overall embryo development. As the ectoderm forms the neural plate and the mesoderm's notochord signals the neural folds to rise, the endoderm creates the internal environment. Think of it as the foundation that keeps the embryo stable while the nervous system develops. Without this support, the other layers couldn't do their job effectively. For step 1, the key point is understanding the division of labor. The endoderm supports the internal environment while the ectoderm and mesoderm builds the nervous system. This concept often shows up in the questions about the germ layer derivatives. For example, a question might ask which layer forms the lining of the gut, which is endoderm, versus the neural tube, which is ectoderm. Knowing these roles helps you tackle those embryology questions with confidence. The endoderm's role in neurulation is less direct, but it's a piece of puzzle you need to know. As we move forward, we will focus on how the neural tube forms and why its disclosure is so critical for the exam. Let's picture the embryo at a pivotal point. Two ridges are about to fuse, creating the foundation of the brain and spinal cord. This is neural tube formation, and it's a high-yield topic for USMLE Step 1 that you need to master. Here is the process. The neural folds, which we saw forming from the ectoderm, grow and fuse at the midline to form the neural tube. This tube will eventually become the brain and spinal cord, the core of the central nervous system. The fusion starts around day 22 and completes by day 28, a critical window in development. Once formed, the neural tube differentiates into the brain at the cranial end and the spinal cord at the caudal end. Why is this so important for step 1? Failure of neural tube closure leads to neural tube defects, which are classic exam topics. If the cranial end fails to close, you get anencephaly, a fatal condition where the brain doesn't fully form. If the caudal end fails to close, you get spina bifida, which can range from mild spina bifida occulta to severe meningomycocele, where these defects are often linked to folate deficiency in clinical vignettes. So you might see a question about a pregnant woman not taking folic acid supplements. Knowing this process helps you identify the risk. Neural tube defects are also tied to genetic and environmental factors, making them a favorite for step 1 questions. So as we move forward into neural crest migration, keep this in mind. Neural tube formation is the foundation of the central nervous system, and its failure has major clinical consequences. Imagine cells embarking on a journey across the embryo, transforming into everything from nerves to adrenal glands. That's the magic for neural crest migration, and it's a high-yield topic for USMLE Step 1. So what are neural crest cells? They are neuroectoderm-derived cells that form at the edges of the neural folds during neurulation. As the neural tube closes, these cells pinch off and migrate in waves throughout the embryo. As we saw in the earlier diagrams, they are incredibly versatile, forming the peripheral nervous system and other structures like the adrenal medulla. A mnemonic to remember this: crest cells wander freely. Crest to cells to migrate. Why does this matter? Neural crest cells are multipotent, meaning they can differentiate into diverse tissues, which makes them a favorite for step 1 questions about embryology and germ layer derivatives. For example, a question might ask which cells form the sensory neuron of the peripheral nervous system. That's neural crest cells. They also contribute to clinical questions. If neural crest migration fails, you can get disorders like Hirschsprung disease, where neural crest cells don't reach the colon, leading to congenital aganglionic megacolon. As we move through the next slides, we will break down the 3 waves of neural crest migrations and the structures they form. This is the key for understanding the peripheral nervous system and other systems, so let's dive into the first wave next. Let's follow the first group of neural crest cells as they race through the embryo like sprinters. This is the first wave of neural crest migration, and it's packed with high yield points for USMLE Step 1. During this wave, neural crest cells form 3 key structures of the autonomic nervous system. First, they create the sympathetic ganglia, which are part of the fight or flight system. Think of them as the relay stations for sympathetic nerves. Next, they form the parasympathetic ganglia, which handles rest and digest functions like slowing the heart rate. Finally, some neural crest cells migrate to the adrenal medulla, becoming the chromaffin cells, which produce catecholamines like epinephrine, basically the core of the adrenal gland. This is a big deal for step 1 because these structures tie directly to the autonomic nervous system, a common exam topic. For example, a question might ask which cells produced epinephrine in the adrenal medulla. That's chromaffin cells from the neural crest. Or you might see a clinical vignette about pheochromocytoma, a tumor of the adrenal medulla, and needs to know its embryological origin. Again, it is the neural crest cells. The key visual here is to think of the first wave as sprinters racing to autonomic roles. As we move to the next wave, we will see how neural crest cells shift to the supporting peripheral nervous system. Keep this in mind. The first wave builds the autonomic framework and its foundation for understanding the peripheral nervous system development on the exam. Now let's follow the second wave of neural crest cells. They are like the support crew for the peripheral nervous system, and this is a critical topic for USMLE Step 1. In this wave, neural crest cells form structures that support the peripheral nervous system. First, they create the posterior root ganglia, which houses the sensory neurons for spinal nerves. Think of them as the sensory hubs for the body. Next, they form Schwann cells, which myelinate peripheral nervous system axons, speeding up nerve conduction. We will dive deeper into Schwann cells on slide 11, but for now, know they're essential for peripheral nervous system function. Finally, neural crest cells become satellite cells, which support neural cell bodies and ganglia, acting like protective cushions. This is a high yield for step 1 because the peripheral nervous system is a common exam topic, especially in questions about the nerve function and embryology. For example, a question might ask, which cell myelinate peripheral nervous system axons? That's Schwann cells derived from the neural crest. Or you might see a clinical scenario about a sensory deficit and need to know the embryological origin of sensory neurons. Those are the posterior root ganglia, also from the neural crest. The key visual here is the second wave as the support crew for the peripheral nervous system, ensuring sensory and supportive functions. As we move forward to the third wave, we will see how neural crest cells contribute to pigmentation and other structures. Keep this in mind. The second wave builds the sensory and supportive framework for the peripheral nervous system, and it's a must-know for the exam. Now let's follow the final wave of the neural crest cells as they add color to the embryo, literally. This is the third wave of the neural crest migration, and it's a high yield topic for the USMLE Step 1. In this wave, neural crest cells formed the melanocytes, which are pigment cells that spread throughout the epidermis. Melanocytes produce melanin, giving color to the skin, hair, and eyes. This process is crucial for normal development, and it's a classic embryology point on the exam as well. For example, a question might ask which cells are responsible for skin pigmentation. That's melanocyte derived from neural crest. Or you might see a vignette about albinism, a condition where melanocytes don't produce melanin and need to know its embryological basis. This wave is also clinically relevant because the neural crest migration defects can lead to pigmentation disorders, which sometimes appear in step 1 questions. For instance, conditions like Waardenburg syndrome, which involves pigmentation abnormalities and hearing loss, are tied to neural crest migration failures. Knowing the embryological origin of melanocytes help you connect the dots in those scenarios. The key point here is that melanocytes give skin, hair, and eye color, and they are product of the third wave of neural crest migration. This wraps up our overview of the neural crest migration, showing how versatile these cells are, from the autonomic nervous system to the peripheral nervous system to pigmentation. For step 1, mastering these waves will help you tackle embryology questions with confidence. Now let's follow the final. Alright, now let's journey to the head and neck of the embryo, where neural crest cells are working their magic to shape the face and sensor systems. This is a fascinating and high yield for step 1. Neural crest cells in the head and neck have unique contributions that you need to know for the exam. First, they form a sensory ganglia for the cranial nerve number 5, 7, 9, and 10. These are the trigeminal, facial, glossopharyngeal, and vagus nerves respectively. These ganglia houses sensory neurons responsible for sensation, like facial, touch, and visceral sensations, making them critical for understanding cranial nerve functions on step 1. Second, neural crest cells migrate to pharyngeal arches, where they create a mesenchyme that forms bone and connective tissue, essentially building the structural framework of the face and neck. This has big clinical relevance. Defects in neural crest migration here can lead to the craniofacial abnormalities, like Treacher-Collins syndrome, which you might see in step 1 vignette. This condition involves underdeveloped facial bones, leading to features like downward slanting eyes and a small jaw, and it's directly tied to failed neural crest migration. A question might present a child with these features and ask for the embryological cause. Knowing the neural crest's role in the head and neck will help you nail that answer. So as we move forward, keep this in mind. Neural crest cells in the head and neck are key players in sensory and structural development, and their failure leads to testable clinical condition. If you're finding this helpful for your step 1 prep, don't forget to like and subscribe for more high-yield content. Picture 2 types of cells wrapping axons like insulation on a wire. One in the peripheral nervous system, one in the central nervous system. This is a difference between Schwann cells and oligodendrocytes, and it's a must-know distinction for step 1. Let's start with the Schwann cells, which we touched on earlier in the neural crest migration. They support peripheral nervous system axons by either cushioning unmyelinated axons or forming myelin around them. As you can see in the diagram on the left, one Schwann cell myelinates a single segment of one axon, and the gaps between Schwann cells are the nodes of Renvie, which speed up the nerve conduction via saltatory conduction. Now compare that to the oligodendrocytes in the central nervous system shown on the right. One oligodendrocyte can myelinate multiple axons. Think of them as a multitasker, wrapping several axons at once to ensure efficient signal transmission in the brain and spinal cord. This distinction is high yield for step 1 because it ties to clinical condition. For example, a question might ask which cells are affected in Guillain-Barré syndrome. That's Schwann cells, leading to peripheral nervous system demyelination. In contrast, multiple sclerosis involves oligodendrocyte damage in the central nervous system. The mnemonic here is Schwann single, oligos many. Schwann cells myelinate one axon segment while oligodendrocyte handles many. The key visual in Schwann cells are individual wrappers versus oligodendrocytes are multitaskers. Understanding this difference will help you tackle neuroanatomy questions on the exam with confidence. If this breakdown is helping you to prep for step 1, be sure to like and subscribe. On the slide 13, you will see the diagrams of Schwann cells and oligodendrocyte. Let's tie it all together with a big picture summary of neurulation and neural crest migration, perfect for locking in those high yield points for step 1. First, neurulation, the ectoderm forms the neural tube, which becomes the brain and spinal cord, while the mesoderm notochord guides this process. And the endoderm provides the GI and respiratory linings. Neural crest cells then migrate in waves, forming the peripheral nervous system, melanocytes, and head and neck structures, as we have covered before. A mnemonic to remember the sequence: neurons crest, tubes connect. Neurulation, crest migration, tube formation. This process is critical because defects lead to clinical conditions. Neural tube defects like spina bifida and anencephaly often shows up in step 1 big necks, tied to folate deficiency. Neural crest defects can cause Hirschsprung disease, where the colon lacks ganglia, or craniofacial anomalies like Treacher-Collins syndrome. These clinical connections are goal for step 1. For example, a question might describe a newborn with the lower back defect and ask for the embryological cause. That's a neural tube defect from failed neurulation. Autovignette about a child with a small jaw and hearing loss points to Treacher-Collins syndrome from neural crest migration failure. My study tip: use mnemonics to keep it fun and memorable as you review. We have covered a lot of the ground today, from the neural plate to Schwann cells, all tailored for step 1 success. If this presentation has helped you, please like and subscribe for more high-yield USMLE content.